You are thirsty in a way that is becoming a problem. Not the mild background thirst of a modern person who has been sitting at a desk for 3 hours and just noticed their water bottle is empty. This is the kind of thirst that has been building for a day and a half, the kind your body is now communicating through every channel it has available. There is a specific dryness in your throat that makes swallowing uncomfortable. A headache that started this morning has been getting steadily worse. The way you are thinking has taken on a fuzziness you do not entirely trust. You need water urgently. This is not negotiable. And there is water right in front of you, roughly 40 meters down a slope. A pool of standing water sits at the base of a rocky outcrop, fed by what looks like a seep or a small spring trickling from the rock face above. It is visibly water. It is accessible. It would solve your immediate problem in every physical sense. But you are not moving toward it. You are looking at it, assessing it, running it through an internal evaluation process that is producing a result somewhere between caution and outright rejection. There is something about it, and you are not even entirely sure what that something is yet, that is making your body say wait before your conscious mind has fully articulated why. This moment, the pause before the water, is the subject of what follows. Because the ability to evaluate water quality without laboratory equipment, without chemical testing kits, without anything beyond accumulated knowledge and trained perception, is one of the most sophisticated and most survival-critical skills that ancient humans possessed. The specific mechanisms through which they knew which water was safe and which water would kill them are both more complex and more interesting than most people ever stop to consider. Start with what contaminated water actually does, because understanding the threat is essential to understanding how ancient humans learned to assess and avoid it. Waterborne pathogens, the bacteria, viruses, protozoa, and other microorganisms that contaminate water sources and cause human illness, are the leading cause of death globally in populations without access to water treatment infrastructure even today. Cholera, typhoid, dysentery, giardia, cryptosporidium, hepatitis A. The list of serious and potentially fatal waterborne diseases is long, and every one of them was present in the ancient world, forming a constant pressure that the human immune system had to contend with across the full span of our species' existence. The disease burden that waterborne illness imposed on ancient populations was significant. Children, with less developed immune systems, less accumulated immunity from prior exposure, and higher susceptibility to the severe dehydration that diarrheal illness produces, were particularly vulnerable. Archaeological and paleopathological evidence from ancient human remains consistently shows signs of chronic parasitic infection and the specific bone changes associated with severe childhood illness, including gastrointestinal infection. This tells us that in the ancient world, water quality was a genuine, serious, frequently lethal concern. Communities that made consistently poor water choices suffered significantly higher mortality than communities that reliably found and used safe water sources. The ability to assess water quality was not a trivial skill. It was survival infrastructure, as important in its domain as fire management or food knowledge. Ancient humans developed it through empirical observation, accumulated cultural knowledge, biological sensory systems calibrated to relevant water quality signals, and the ecological intelligence that grows out of sustained observation of the relationship between water sources and the health outcomes of the people and animals who use them. Consider the sensory systems humans have specifically for water quality assessment, because evolution did not leave ancient humans without biological tools for this problem. The human sense of smell is significantly more capable than most people appreciate in the specific domain of detecting water contamination. The olfactory bulb, the brain structure that processes smell, connects directly to the limbic system, the set of brain structures involved in emotional and motivational processing. This direct connection, which bypasses much of the cortical processing that most sensory information undergoes, is the reason smells produce such immediate emotional responses and are uniquely effective at triggering avoidance or approach behavior. The specific chemicals that produce the characteristic odors of water contamination, hydrogen sulfide from anaerobic bacterial activity, various organic compounds from algal blooms, ammonia from nitrogen-rich animal waste, carry distinct chemical signatures that different categories of microbial contamination reliably produce, and the human olfactory system can detect them at concentrations relevant to actual contamination risk. The threshold concentrations at which many of these chemical markers become detectable to humans fall in roughly the same range as the concentrations at which they begin to indicate genuine contamination risk. This is not a coincidence. It reflects millions of years of selection acting on the primate olfactory system in an environment where contaminated water was a genuine mortality risk. Humans, whose olfactory systems could detect the relevant contamination signals at relevant concentrations, survived and reproduced at higher rates than those whose detection thresholds were too high to be useful. Over many generations, the human sense of smell was tuned, specifically and functionally, to the chemical markers of water contamination. The disgust response that bad-smelling water triggers, the automatic, immediate, physically strong aversion most people feel when they encounter the smell of contaminated water, is the motivational complement to that olfactory capability. It is the behavioral expression of the neural link between olfactory detection and limbic aversion. The smell is detected, the limbic system produces a strong negative emotional response, and the behavioral result is avoidance. This is exactly the biological circuit you want when the thing being detected is genuinely dangerous. Visual assessment extends this system well beyond smell into the detailed visual analysis that the human eye and brain are specifically equipped to perform on complex natural stimuli. Color is one of the most immediate and informative visual water quality signals available. Clear, colorless water is not a guarantee of safety, but turbid, colored, or otherwise visually distinctive water provides specific information about the conditions that produce those characteristics. A green or blue-green discoloration in standing water indicates an algal bloom, potentially including toxic cyanobacteria that produce compounds harmful to humans and animals alike. Red or reddish-brown discoloration can indicate iron contamination or the biological blooms associated with it. Milky or white turbidity often signals mineral precipitation or suspended particulate matter. Black or darkly discolored water indicates a high organic matter content and the microbial activity associated with decomposition. None of these color signals functions as a definitive diagnostic test in the way modern laboratory water testing does, but each one is a genuine signal. They encode real information about the conditions of a water source, information that ancient humans could read and interpret within a framework of accumulated experience that told them what those signals tended to predict about health outcomes. Ancient humans had observed, over the course of individual lifetimes and across the transmitted knowledge of many generations, that people who drank from green-tinged pools got sick, while people who drank from clear springs generally did not. Those observations, repeated across enough people and enough time, amounted to a functional, empirically validated, and culturally transmitted body of water quality knowledge. Clarity carries information beyond color as well. Water that is turbid with suspended particles, even particles that produce no visible color change, has usually been recently disturbed, or flows through soil conditions that produce heavy sedimentation. Turbid water also tends to carry a higher pathogen load than clear water, because the surfaces of suspended particles offer attachment sites for bacteria. Ancient humans who had learned, through repeated experience, that turbid water from certain kinds of sources more often produced illness than clear water from similar sources had derived a functional water quality heuristic that modern water science independently confirms. The single most important water quality signal available to ancient humans, though, was the behavior of animals. Animals are the most powerful and most reliable water quality indicator that exists in a natural landscape, and ancient humans, whose ecological intelligence included extraordinarily detailed knowledge of local animal behavior, were reading this indicator constantly and skillfully. The logic behind it is straightforward. Animals in a landscape have been using the same water sources for generations. They share, broadly, the same biological vulnerability to waterborne pathogens that humans have, sometimes greater, sometimes different in specifics, but comparable in severity. Water sources that are reliably contaminated produce consistent illness and death in the animals that use them, which in turn produces consistent avoidance of those sources by the healthy animals remaining in the local population. Water sources that are reliably safe are reliably used by that same healthy population. An ancient human who observed deer, horses, or other local wildlife gathering freely at a particular water source, drinking without hesitation and showing no subsequent signs of illness, had strong evidence that the source was safe for human consumption as well. An ancient human who observed a water source with no animal use at all, no tracks in the mud, no evidence of recent animal presence, despite the local animals clearly needing water somewhere, had equally strong evidence that something about the source was unsafe. The distribution of animal tracks, the frequency with which different water sources were used, the visible health and behavior of the animals drinking there, all of this constituted environmental information that ancient humans read continuously as part of the ordinary business of navigating their territory. This is not intuitive guesswork. It is rational inference drawn from a remarkably well-developed monitoring system. The animals of a landscape had already been running the empirical test on local water sources continuously, with their own health as the outcome measure, for far longer than any human community had been present in that place. Their collective behavioral evidence about which sources were safe and which were not was more extensive, more locally calibrated, and more continuously updated than any human community's direct experience alone could have produced. Ancient humans who understood this were, in effect, using the animal population of their territory as a distributed water quality testing network, because that is exactly what it was. Beyond the behavior of animals, ancient humans developed detailed knowledge of the landscape features associated with water quality, knowledge that was functionally equivalent to modern hydrogeological understanding, even without the vocabulary or instruments of modern science. Moving water is generally safer than standing water, and this is a well-established principle that reflects real biology. The flow of moving water dilutes pathogen concentrations, denies many pathogen life cycles the residence time they need to complete, and creates conditions of aeration and temperature variation that are unfavorable to many pathogenic organisms. Ancient humans who preferentially sought out moving water over standing water were applying a rule that modern water quality science independently confirms. The source of the water matters enormously as well. Groundwater emerging from rock or deep soil as a spring has passed through geological media that filter out particulate matter and many biological contaminants, offering real protective benefit. Rainwater collected before it touches the ground is free of the soil-derived microbial contamination that water carries once it makes contact with the earth. Surface water in areas of high animal density tends to carry a significantly higher pathogen load than surface water in areas where fewer animals congregate. Ancient humans with detailed knowledge of their own territories carried a kind of spatial map of water quality in their minds, encoded in the accumulated knowledge of their community across generations. They knew which springs were reliably clean, which streams turned unsafe at particular times of year when upstream conditions shifted, which pools filled after rain and were safe to drink from while fresh, but grew dangerous after several days of standing, and which parts of their territory carried surface water that was reliably contaminated by seasonal concentrations of animals. This spatial water quality map was never written down anywhere. It existed in the minds of a community's most experienced members, and was passed on through the specific oral traditions that preserved survival-critical knowledge across generations. The elder who said, "Do not drink from the eastern pool in the dry season," was transmitting a water quality assessment that had been validated through generations of empirical observation, even if the precise mechanism, what exactly made the eastern pool dangerous in the dry season, was understood in ecological terms rather than microbiological ones. Assessment and avoidance were not the only tools available to ancient communities managing the risk of contaminated water. There were also active practices for making questionable water safer to drink. Fire and boiling represent the single most effective water treatment technology available without modern infrastructure, and there is good reason to think ancient humans understood and used this technology wherever it was accessible and necessary. Boiling water produces immediate and effective destruction of bacterial and protozoan pathogens, significantly reduces viral load, and eliminates the categories of threat that represent the most immediate waterborne illness risks. The knowledge that boiling water made it safer, that people who drank water heated over a fire were less likely to fall ill than people who drank the same water raw, is exactly the kind of empirical observation that ancient communities with fire technology had both the capacity and the motivation to make, given how much was at stake. Whether ancient humans understood why boiling worked is a separate question from whether they observed that it worked. The ethnographic and documentary record of traditional communities around the world consistently shows a practice of preferentially boiling water whenever its quality is in doubt. Establishing the antiquity of this practice archaeologically is difficult, since the behavior itself leaves no physical trace distinct from other everyday uses of fire, but the combination of available fire technology and the sheer necessity created by waterborne illness makes it a plausible and likely ancient practice, with strong supporting logic even where direct evidence is scarce. Plant-based water treatment is a separate and genuinely fascinating dimension of ancient water safety practice. Multiple plant species produce compounds with antimicrobial properties that, when added to water, measurably reduce pathogen loads and lower illness risk. Certain barks, seeds, and roots have served as water treatment agents in traditional communities across Africa, Asia, and the Americas for a very long time. Moringa oleifera seeds, which contain specific proteins that cause bacterial cells to clump together and settle out of suspension, have been used as a water clarification agent in African traditional communities, and their effectiveness has since been confirmed by modern water treatment researchers working in laboratory settings. Other plant species with similar antimicrobial or clarifying properties appear again and again in the traditional water treatment practices of communities across the world. Ancient humans possessed detailed botanical knowledge that was deep, specific, and empirically grounded, and they had both access to these plant resources and the observational framework needed to notice their water treatment properties. The connection between adding a particular plant material to water and seeing improved health outcomes among the people who drank it afterward is precisely the kind of cause and effect observation that ancient empirical knowledge systems were built to make and to retain. Taste played its own role in ancient water quality assessment, and this is more interesting and more genuinely functional than it might first appear. The human taste system includes specific receptors sensitive to several of the chemical markers associated with water contamination. Sulfur compounds, which indicate anaerobic bacterial activity, register as bitter or sulfurous even at low concentrations. High mineral content, which can point to specific geological contamination sources, produces distinctly mineral or metallic tastes. A high organic load produces earthy, musty, or putrid tastes, depending on the particular biological community responsible for it. The bitter taste response in particular, one of the most immediately aversive taste reactions in humans, shared in its basic properties across every human culture, is specifically calibrated to compounds that are frequently toxic. The bitterness that shows up in water from certain contaminated sources is often produced by the same chemical compounds, algal toxins, mineral contamination, or the metabolic byproducts of pathogenic microorganisms that make the water dangerous in the first place. The bitter taste response functions as the body's last line of sensory defense. If something slipped past the nose and the eyes, the tongue may still catch it. Ancient humans who had learned to trust that aversion, who had noticed, or whose community's accumulated knowledge had encoded, that water tasting wrong more often led to illness than water tasting fine, were relying on a genuine, functional diagnostic tool. It was not infallible. Some dangerous contaminants are essentially tasteless, and some unpleasant tastes come from entirely harmless compounds. But it was better than chance, and far better than no assessment at all. The training of the taste system, learning which specific taste qualities tended to be associated with which specific outcomes, was part of the accumulated experiential knowledge that ancient communities passed down across generations. The child who was taught that water tasting like this is not safe to drink was receiving empirically validated guidance, delivered in a form that was memorable and immediately actionable. Return, now, to the pool at the start of all this. You are still looking at it. Your thirst has not eased. The water is still sitting there, 40 meters away, exactly as accessible as it was a few minutes ago. But in the pause since you first noticed it, your evaluation has been quietly running its course. The smell is wrong. Not dramatically wrong, not the overwhelming sulfurous stench of heavily contaminated water that would send anyone running. Subtly wrong. There is a quality in the air near the pool that your olfactory system has registered as not right, in a way that is producing a mild but persistent aversion. You did not consciously decide to think about the smell. Your nose simply told you something, and some older part of your brain listened. The color is slightly off as well. A tinge that is barely perceptible in the afternoon light, but that, looked at carefully, is not quite the colorless clarity of safe water. Something faintly greenish sits at the shallow margins, and there are no tracks at the edge. This is the detail bothering you most. You can see the mud along the water's edge clearly, and it is smooth. No hoof prints, no paw marks, no bird tracks in the wet ground. For a water source in this landscape, in this season, the absence of animal use is itself a piece of information, and a significant one. Whatever the animals of this place know about this particular pool, their absence is telling you the answer without a single word passing between you. There is a spring 2 kilometers to the east, uphill, where the rock changes from dark sedimentary stone to lighter granite. You have been there before. It means 2 more kilometers of thirst on top of what you are already carrying, but the water there runs clear and cold, and the ground around it is tracked by every animal in the area, and it has been safe every time you have used it. You turn east. That decision, made without a laboratory, without a pH strip, without any technology beyond accumulated knowledge and trained perception, is the ancient water quality assessment system doing exactly what it evolved and was built to do. And it works. Not perfectly, not every single time, and not without the occasional catastrophic error that taught the next generation a hard lesson about what not to do. But it worked reliably enough, across enough generations of accumulated and transmitted knowledge, that ancient humans managed to keep themselves adequately hydrated across the full range of environments their species came to inhabit. They looked at water the way they looked at everything else around them, as a complex system carrying specific signals, readable with the right knowledge, accessible with the right trained perception. They built those skills across generations of careful observation and costly, sometimes fatal, experience. And they encoded what they learned into cultural knowledge that persisted, accumulated, and remained available to every generation of their community that came after. They knew which water was safe. Not perfectly, not always, but reliably enough, practically enough, to survive.